So my name is Guillaume Lesage. I'm the treasurer of the CNSP and a co-organizer of this event. I'm also working at McGill as a grant officer in the Office of the Vice Principal, Research and Innovation. So it's my pleasure to be here today to moderate this session highlighting the success of the Quebec COVID-19 Biobank, la Biobank québécoise de la COVID-19, referred to as the BQC-19. So when we started to prepare this virtual event, it was obvious to me that the BQC-19 should be presented as a success story. I mean, this scientific platform was created last year as a response to the pandemic and became operational very fast and despite very challenging times. As soon as the project was initiated in the spring 2021, several administrative units at McGill were engaged to coordinate and support the setup of the biobank. For my part, I've been involved in the participation and negotiation of the many agreements that regulate the activities of the biobank. And we'll describe this in a few minutes. But the presentation that will be given now will be given by three speakers. First, we'll hear Vincent Moser, who's the director of the Quebec COVID-19 Biobank. Vincent is a physician scientist. He holds the Canada Excellence Research Chair in Genomic Medicine at McGill. He came to McGill in 2019. Before moving to Canada, he held various academic positions in Switzerland, including chair of the laboratory department at the Lausanne University Hospital and vice dean for clinical research at the Faculty of Biology and Medicine at the Lausanne University. Vincent also spent 10 years running R&D in the industry at GlaxoSmithKline, so JSK, in Philadelphia, as vice president in charge of applied genomics and as the head of genetics for rare diseases. The second speaker is Pascale Léon. So Pascale is the manager of the Quebec COVID-19 Biobank. She received her PhD in life and health sciences from the Université de Paris 6 in France. After a postdoc in the US, she moved to Canada and worked for a few years as a scientist at the University of Toronto and then at the University Health Network. Since 2004, she has had several executive positions in the management of large research endeavors at the Ontario Institute for Cancer Research in Toronto, and then at the Canadian Consortium on Neurodegeneration in Aging at the Lady Davis Institute for Medical Research in Montreal. And then the last part of the presentation will be given by myself. Following the presentations, we will have a session of questions and answers as a panel. So we encourage you to ask your questions in the Q&A box at the bottom of the Zoom window, and we will answer them after the presentation. The title of the presentation is The Quebec COVID-19 Biobank as a Unique Research Platform to Study COVID-19. And to start the discussions, I would like to invite Vincent Moser.
Oui, bonjour à tous. Thank you very much, Guillaume and the organizers, for giving us, Pascale and myself, the chance to describe this platform in this very interesting meeting you have organized. So specifically here, we will focus on one platform, which is called the Quebec COVID-19 Biobank. The rationale for building up this biobank is shown on the next slide. On the left-hand side, you can see sort of the distribution of how people react when they have been exposed to SARS-CoV-2, the virus responsible for COVID. In green, you can see that the vast majority of people are essentially asymptomatic or have a mild disease. Some people in yellow have a symptomatic disease, fever, fatigue, loss of smell. Some people end up in the hospital with severe cases and breathing difficulties. And some of them, unfortunately, die, a fraction of them, probably 1 or 2%. And the question we're really asked to try to address is the following one. That's the same virus, SARS-CoV-2. So why do people react so differently to exposure to this virus? And this is not a trivial question. It's an important question because if we can better understand why some people react badly, for instance, we could imagine diagnostic tests to identify early who is at risk of developing severe diseases. And that can have immediate application. Imagine, for instance, a patient coming in the ER is coughing. If a test can tell, wow, be careful, this person will have a very severe outcome, then it's worth it to book a bed in the ICU for this person. If this person is deemed to have a favorable outcome, then it means that this person can go back home. So this is important in terms of diagnostic and, of course, in terms of treatment. As you know, there are limited treatment opportunities for this disease. And so new treatments are needed, even if, luckily enough, vaccines are now taking care of part of, at least, the problem. So the next slide shows you here the mandate we received from originally the provincial government here in Quebec. It was really the chief scientific officer and the head of Génome Québec and the head of the Fonds de Recherche Québec en Santé who gave us this mandate, build up a platform, an infrastructure which will support research to identify the determinants of COVID-19 susceptibility. That means, why do some people develop a disease? Severity, why do some people end up, for instance, in the ICU? And outcome, why do some people, for instance, have what we would call now long COVID? That means sequelae of the infection. In that sense, and that's where PHAC came in, the Public Health Agency of Canada, they wanted us to contribute to public health response to the COVID-19 pandemic. The next slide summarizes what we have done in a month. And we have put in place a steering committee of 10 people with various expertise, be it finance, genomics, ethics, legal, immunology, virology, and so forth. We designed the study, which I will describe to you later. We put a governance in place. We got part of the funding. The other part came later with PHAC. We wrote up the protocol and the SOPs. We got a fixed ethics approval to start the recruitment. And we also put in place the IT infrastructure to collect the data and have the samples properly identified. All that stuff was done within 10 days, actually. We received the mandate on 21st of March. Our first patient was recruited at the Jewish General Hospital here in Montreal 10 days later on April 1st. And that next slide, I think, by itself is a world record. Normally, outside COVID, you probably need a year to put that in place. Here, because of the vision, because of also the willingness of the stakeholders to get something done, but also, and Pascale will come back to that later, because of the existing platforms, it was really possible to do that. And that's really what I want to show in the next slide. All these things could happen because existing platforms. Now, I want to describe to you the study design. That's the next slide. Again, keeping in mind the mandate we received, that means finding clinical and molecular determinants of variable susceptibility, severity, and outcome of COVID, we designed this study as such. It's a prospective study with serial measurements over a 2-year period. It's an observational study where we collect extensive data plus blood at each visit. It's multi-centric across the province with 9 centers recruiting, affiliated with 5 major Quebec universities. So I think by itself, that's a tour de force to put all these people together. And here I express my gratitude to Guillaume and his team for making it feasible. It's broad. We are collecting individuals who are asymptomatic and patients who are severely ill. It's designed what we would call a case control. That means the capacity we have to compare people who are SARS-CoV-2 positive on PCR with people who are SARS-CoV-2 negative. And it's collaborative. We are not working in silo. We want here to be hooked with other initiatives like Concogen, CITF, CONCOV, and others, but also having the chance to collaborate internationally, because the challenges we are facing are beyond what one single lab or province can really address. The next slide summarizes what we were able to do within a year. We have recruited 2,800 participants, now 3,500. That's almost 7,000 visits. For 2,000 of these visits, we have what we call a multi-omic profiling on the samples collected during this visit. That means a very detailed molecular assessment of the samples we have collected. This includes, for instance, sequencing of the host genome, measuring 5,000 proteins in each sample, measuring the anti-SARS-CoV-2 antibodies, measuring as well a variety of other parameters and small molecules. This is a platform again, and our goal is not to accumulate data. It is to share data after we have generated them rapidly and with high quality. So we have put in place some national and international collaborations, and so far we have contributed to 7 publications published or preprint. The next slide summarizes to you what are the numbers of patients we have recruited. On the left-hand side, you can see patients who are severely ill. In blue, people who are cases, that means SARS-CoV-2 positive. In green, those who are controls, negative. And on the right-hand side, you can see the number of people who we have recruited with less severe disease or asymptomatic, be it PCR positive or controls PCR negative. So by itself already, we have one of the largest collection of patients properly recruited and consented for studying at molecular level the determinants of COVID. The next slide illustrates to you one piece of data. This is the results from what is called a genome-wide association study, or GWAS analysis. In this type of analysis, what we do is we interrogate variants spread along the human genome. Each of these variants here, and there are 2 million of them on this slide, are depicted by a dot. The location of the dot along the chromosomes, that means in the genome, is shown on the x-axis. For each of these 2 million dots or SNPs, variants, we ask the question, is this variant associated or not with COVID, severe COVID versus controls? At the bottom, you can see the number of people who had undergone these analyses, 12,000 people with severe COVID, a fraction of them coming from a biobank, the other one coming from a variety of laboratories around the world, and 1.7 million controls. And what you can see here on this called Manhattan plot is a series of towers. That means regions in the genome which are associated with COVID severity. Under the arrows, you can see on the left-hand side a peak in chromosome 3, and on chromosome 12, another peak with the label OAS1. I will give you now more information on the variant on chromosome 3, which is the next slide. At the bottom of the left-hand side, you can see the number of people who carry this very variant. You can see that 8,000 people do not carry the variant, 1,400 do carry the variant. So altogether, 14% or so of the population does carry this variant. On the x-axis, you can see the cumulative risk of ending up in ICU or dying from COVID, according in yellow to the presence of the variant. Yellow people do not carry the variant. Blue depicts the 14% of the population who carries this variant. And what you can see is that carriers of variant have a 40% increased risk of ending up in ICU or dying from COVID-19. So essentially, we have reached one of these goals. We have identified a genetic marker associated with outcome. On the right-hand side, you can see what is the risk associated with the presence of this variant and compare that with other comorbidities: diabetes, DM, congestive heart failure, COPD, chronic kidney disease, cancer, obesity, and so forth. And what you can see is that the carriers of this variant have a risk, genetic risk, as much and as pronounced as the one that people who have diabetes or COPD or chronic kidney disease. So it's an important risk factor. And the question is now, shall we measure this variant in people so as to stratify their risk? As you remember, the next slide refers to the other tower on chromosome 12, the one associated with OAS1. OAS1 is a protein which is expressed by this gene. What Brent Richards at the Jewish General Hospital here in Montreal and a very large team of investigators is they have analyzed the function of OAS1. And in the next slide, they have demonstrated, the previous slide, that this protein does protect from COVID. And it may be then a new therapeutic target for COVID-19. So in summary, we have put together this platform. It's been very hard work. We're very glad we could have the chance to build it because of the vision and funding of our sponsors, thanks to the availability of existing platforms. And we look forward to continuing this big effort and making the data and samples available to the entire community. Thank you. Now, please, Pascale, the floor is yours.
Hi, everyone. Can you hear me? Okay. So in support of all these wonderful research results that, that Vincent just shown, there are a lot of, as you can imagine, a lot of work that's going on behind the scene. And I want to touch up briefly on two aspects: the governance of the BQC19 and all the platforms that are used to facilitate access to our samples and data, which are, as Vincent mentioned, really at the heart of our mission. So we do rely, I apologize for the typo, so the BQC19 relies on many platforms. These are just a few of the platforms, but they are very important ones. Of course, Nagano for everything that is ethics related, RedCap for everything that is related to databases. Our data are stored in platform under Calcul Quebec. The biobank of the RSRQ in Quebec City also helps with, is used for the databases related to inventories of the samples. We have a collaboration with the National DLML in Winnipeg, the National Microbiology Laboratory, which is financed by the Government of Canada. And then the whole genome sequencing is done at the McGill Genome Center. And that's the place also where all the samples are centralized before being then dispatched to the different laboratories that will do are doing the what we call the core analysis, is all these different kinds of analysis on 2,000 samples that Vincent just mentioned. So in terms of governance, this is the organigram of how the BQC19 is organized. So director, the steering committee is what Vincent mentioned was assembled in record speed, a group of about 10 people with different expertise. On the right-hand side, you can see the staff support. So it's a very lean organization with myself as the manager. We have an access officer who deals with, who oversees everything related to access to samples and data. And a very important role also is played by our coordinator, who is located in Quebec City, and deals with everything related with the databases, which are really at the heart of the BQC19. Importantly, we have also in the first year assembled fairly quickly an international scientific council. And this is a group of international experts, very high caliber, that advise us on strategic direction for the BQC19. They meet every few months and give us very valuable insight of what's happening in the world, but also where we should go in terms of where we could go in terms of strategic plan. This is a group of experts in all sorts, again, of areas, from ethics to genomics to public health and epidemiology. At the bottom of the slide, you can see all these different areas of expertise. We call them dicastères in French, where that is represented at the steering committee level. And each member of the steering committee is responsible for one of these areas, one of these dicastères. So access data samples, the operation, the databases, etc. So the communication, psychosocial, pediatrics, ethics, legal, etc. So it's a very diverse group of experts and Vincent forgot to mention, I think, that we actually, when they started the BQC19 last year, they met every day, including Saturday and Sunday for a few months. And then now, thankfully, thank God, we're down to one meeting a week, but we've been meeting every Thursday morning to organize things. So like Vincent mentioned, the core of the mandate that was given to us for the BQC19 is not simply, of course, to collect samples and store them, but to facilitate access to this BQC19 data and sample for the entire world, basically for the entire scientific community, not just in Quebec, not just in Canada, and not just academic, but also private companies. So the guiding principles around the access is to ensure that scientists have access to the biological material and data for the research efforts on COVID-19. And a very important point is that we being, let's say, Vincent or me or the steering committee, are not the one deciding on who has access to what. This is a very important aspect of the BQC19, but on the contrary, access to data and sample is under the responsibility of an independent access committee who is really solely responsible for granting access to data and sample. So we have a process in place which we facilitate, but which we do not tamper with or intervene in. And this all hinges around this independent access committee. So to date, we have, this is the situation in terms of data and sample access. We're really hoping to ramp up. We've been doing a lot of improvement to our processes around data and samples. And as we go along, as you know, with every platform, you learn from the previous cycle of access, and each cycle of access allows us to make significant improvement to our process. So in terms of data, we've granted access to 14 of our provincial collection, which represent the collection for all hospitals, and then 56 projects have been granted access to data in terms of the local collection that the hospitals have. And then in terms of samples, we're a little lower. We've had only 9 requests and 4 have been granted, but we are now embarking on a major communication strategy to really make the BQC19 better known and make the scientific community know that we have this wonderful resource that, that is in place. So this is just a snapshot of what has been happening, and I'll now turn it over to Guillaume for all the administration.
So thank you very much, Pascale. So yes, when I invited Vincent and Pascale to give this talk, Vincent turned the invitation back to me saying, well, maybe you should present something about the administrator's role in setting up the BQC19. So that's how I came with this short presentation, the administrator's perspective. And in fact, as Vincent and Pascale showed, scientific platforms can usually be described at the scientific level with a great science and great questions to be solved and the great tools to solve those questions. So that's what Vincent presented. And they can also be described at the operation level. That's what Pascale did, showing the management team and the committees and the structure of the BQC. But well, Pascale, stole my punch, but she said that some things do happen behind the scenes. So that's exactly that was exactly my point. That at the administration level, some things do happen also behind the scene. So I mean here that the administration and the institutions do support the scientific platforms. And that's why I have put, as you can see at the bottom of the slide, the McGill logo close to the BQC19 logo, just to strengthen and highlight this support. In the McGill administration, 3 units are involved in this project. So there are some teams in the Office of the Vice Principal Research and Innovation, some people in the legal services, and teams in financial services. And these 3 units are involved in 5, what I would call 5 key administrative areas, which are a liaison with the funding agencies, a liaison with the partners institution, the financial matters, the material data transfer agreements, and the preparation of service agreements. And we'll go a bit more in more details in all of these, in each of these areas. On the next slide, please. So first, the liaison with the funding agencies. So our first role, the first role from the administration is to interface with the research team to validate the budget. As Vincent mentioned, the budget for the BQC19 has been set up in 2 stages. There was first a provincial involvement of $3 million budget, and then a second injection of money from the federal government for $7 million. So we had to build this budget by discussing a lot with the researchers, or a lot with Vincent and all the teams involved, to make sure that that budget was well set for 2 years to cover the salaries of the people, to cover the processes of sample collection, the way to transfer money to the collection sites, for the sequencing, for the additional analysis. So that budget was not an easy task to set. Then the administration has also been involved in preparing award agreements with the funding agencies. Again, the funding is coming from the Public Health Agency of Canada, the federal level, and there was a second agreement that was made with the provincial agencies, the Fonds de recherche du Québec en santé and Genome Quebec. And this is not a recurring agreement. Of course, these there's no recurring competition for that. It's a one of its kind agreement. So the terms of the agreement had to be negotiated with the funding agencies, and that was some not tough, but some job and work from the administration. And then the administration, of course, is involved in ensuring and coordinating the reporting with the funding agencies and make sure everybody is in touch with the PHAC and FRQS and Genome Quebec on time. Second, there's the liaison with the partner institutions. So first, at the onset of the biobank, we had to negotiate an interinstitutional agreement with many institutions. again, there were in this agreement 13 institutions involving 45 parties. So this interinstitutional agreement is very important because it regulates and coordinates the collaboration between all these institutions, and it regulates the sharing of the material and data, and of course the sharing of the funds. So it was kind of, as Vincent said, a tour de force to have this interinstitutional agreement set during last year. Now we are in the process of amending this interinstitutional agreement because additional institutions want to join the BQC19. Administrative people are also involved in their participation to the governing committee described by Pascale, and more specifically, the Vice Principal Research and Innovation and the Deputy Vice Principal in Research and Innovation are involved sitting on this committee. And then lastly, the administrative people are involved also in verifying the ethics compliance and helping with the management frameworks. So the management framework is this document that that regulates all the activities of the BQC19, and it's part of the ethics package. And there's a team in the Office of the Vice Principal Research Innovation who is involved in reviewing the ethics document. And there's a lot of communication between the Research Ethics Board from McGill and the Research Ethics Board from the Centre Hospitalier de l'Université de Montréal, because most of the ethics documents are deposited at the CHUM. On the next slide, please. For the financial matters, of course, this area involves a lot of financial services. So people from finance were involved in opening the funds. So all the financial transactions. Before the award agreements were signed, McGill had to advance some funds to allow the project to start. So financial services were involved in this. And then once the award agreement have been signed and the money has been received, then they are of course managing the deposits. Financial people are also involved in overseeing the transaction and make sure that the transactions comply with the McGill policies and the funding agency policies. So they oversee all the purchases, the salaries, the payment to the sites, and the payment to the service providers. And lastly, finance people are involved also in the financial reporting. And I regrouped the two last points into a single area, a single administrative area, because these are all talking about writing agreements. So there's a material data transfer agreement and the service agreement. Our office and administrative people were involved in setting up the original material and data transfer agreement, so the template that is now used, as Pascale mentioned, by the access committee to grant access to the samples and the data hosted by the BQC. And as Vincent mentioned, there are some multi-omics analysis, and what Pascale called the core analysis. So these are downstream analysis on the samples from the BQC19. This can be immunological analysis, transcriptomics, sequencing, proteomics analysis. And this is done, this is regulated through service agreements that have to be written, and we're involved in this process too. And this is involving people at the CHUM, so academic laboratories, the IUCPQ, and facilities at the IUCPQ, which is l'Institut Universitaire de Cardiologie et de Pneumologie du Québec at Quebec City, and for example, the National Microbiology Lab in Winnipeg, which is a federal facility. So just to conclude that, as you see at the end of this short presentation, the institutional support can take many forms. Of course, we're not working full-time on this project, but there's a lot of support to help the project fly. And I highlighted a lot what McGill is doing. And of course, in other institutions, there are a lot of administrative people who are also involved in helping and supporting the BQC19. I would like to conclude by acknowledging all the people who worked on this project. So first, the BQC19 participants, which are the patients who gave samples, but also the control person who are not actual patients. Of course, we want also to thank the funding agencies, PHAC, FRQS, and Genome Quebec. We want to thank people from sitting on the steering committee and on the governing committees, because this is taking a lot of their time and they are directing this platform very well. And lastly, there's a lot of people who contributed to the success of this platform. So people who are nurses, people working on the collection side, people working to recruit the patients, researchers, technicians, administrative people who worked in the recruitment centers and in several universities. Then next slide, which is the end of the talk. And so I think that now I will thank you everyone for your attention.